I am more than honored, I am humbled to welcome via real-time internet link to the Wired Health stage in London, Craig Venter.
Well, nice to be with you this afternoon or morning in La Jolla. I understand Wired has trouble calculating time differences, but that's all right. I got a call while I was in the shower saying, Wired miscalculated and you're on in 20 minutes. So, fortunately, I live near to human longevity and have a fast car. So anyway, I'm delighted to be with you guys. And the picture you're seeing on screen, if the slides are up, is our headquarter building here in La Jolla. And also in La Jolla is the Venter Institute campus on UCSD site overlooking the ocean. This is the world's first carbon neutral research building and has won lots of recent architectural awards. So it does basic research, synthetic genomics and human longevity, try and convert that into some real world applications. Now, I think most people are aware that the first genome was sequenced in 1995, and that was using a new approach of using new algorithms to quickly assemble genomes. We scaled that up substantially, but the key questions we've been trying to ask and answer from the beginning is, does DNA code for everything in life? And we've taken a lot of approaches to it. One was part of the introduction is synthetic life, where we spent about a decade learning how to write the genetic code till we could get to a 1.1 million base pair code that we booted up in 2010, resulting in this article in Science. That was sort of the control experiment. Since then, we tried designing an organism from scratch. It took another 5 years to do that, as the key message is about a third of the essential genes for life were missing from the scientific repertoire. For 20 years, people have been looking at the same subset, convincing them that they had a set of about 250 to 300 genes, but it wasn't until we actually tried to build a cell and have a living cell from a defined gene set that we found that about a third of the genes were missing. This is essential as we're trying to understand the human genome. This is actually a picture of synthetic 3.0, which is actually the first organism designed in the lab. It doesn't do anything spectacular except make lots of copies of itself, but it does it with this minimal gene set. And now that we have these 149 genes of unknown function, and then they're essential for life, they're helping now to search out those functions. With human, we have a much bigger challenge than just trying to understand a minimal cell, because we have over 20,000 genes and we don't know the function of most of them. But we do know from the synthetic genomic work that we do have DNA software, and that controls every one of our biological functions and codes for our structures. So understanding that language is key to understanding human biology. We did the first complete diploid genome in 2007, and that's the first time we saw the full 6.2 billion base pairs of human life, and found out that each of the genome sets that we get from our parents are substantially different from each other, and that we have things called compound heterozygotes with two different protein structures emanating from the same gene source. We decided the only way to progress was to scale up, not from 1 or 2 genomes, but to hundreds of thousands to millions of genomes. So 2 years ago, we formed Human Longevity, set up the world's largest sequencing facility, and thus far, we're sequencing a human genome roughly every 15 minutes to high coverage for about $1,500. So Compared to where we were in 2000, that first genome cost $100 million. Took 9 months to do with 2 buildings full of sequencers and computers. So there's been big changes over this time period, but the definition of genomics seems to have changed quite a bit, and it's kind of like political language in the US. These definitions have become very fungible. Recently there was a paper in Nature describing the sequence of 1,000 human genomes. It turns out it was sequenced from 1,000 human genomes. It wasn't 1,000 human genomes. If you look at the left line on this, those genomes were covered less than 30%. And so it was serial sequencing. We sequence genomes to 30x coverage, which gives us 10 sequences at least for every sequenceable base pair in the genome. And for having clinical diagnoses, really accurate data is really essential. This map shows that we're still not covering 100% of the genome. The sequenceable genome is about 85%. These are these blue areas. The gray areas, for example, centromeres and other areas with highly repetitive DNA don't yield highly accurate sequence, but fortunately there's no genes in these regions. And you can see with our degree of coverage, our false positive and false negative rates are incredibly low, making this now highly reliable for clinical sequencing. About 10,000 genomes, we saturated the common variants in the human population. And now we see just extremely rare variants. If we were to sequence any of one of your genomes, we'd come up with about 8,000 new rare variants. We have over 100 million now variants in the genome that only occur in our database one time. And it's these rare variants that are essential for understanding for diseases and linked to traits, not the common variants that 23andMe and others measure on their chips. We're applying this highly accurate sequencing to a lot of different areas. For example, with cancer, we have a comprehensive cancer program where we sequence the full genome of the patient, the full genome to 90x coverage on the tumor. We sequence RNA in the tumor. We isolate T cells from the tumor and sequence the entire T cell repertoire. We do liquid biopsy assays for circulating DNA in the bloodstream and put all this together in an accurate review of somebody's cancer and where to go next. For example, with this, we get a full tumor burden. In this case, it was a head and neck tumor. There are 25,000 mutations across the genome, 315 of these in protein coding regions creating neoantigens. We found 4 sites where HPV inserted in the genome, disrupting genes leading to cancer. This individual had several oncogenes that were mutated in his genome, as well as genes associated with immune suppression. And from the microbiome, we found HPV-16 present. So that combination alone would probably mean when, not if, this individual would get cancer. Looking at the tumor, we can find sites that are mutated that drugs work on. But the next stage that we're working on is a specific RNA vaccine for each individual designed around the neoantigens and their cancer. We're treated the first patient only. In this case, we isolated the T cells from the tumor, selected out with peptides made from the neoantigens, the ones that actually attack neoantigens, drew those up and gave those back to the patient to kill his tumor, and thus far it's been very effective. So the key information you get from sequencing, aside from druggable targets, are the neoantigens that create all kinds of new therapeutic approaches, and I think this is one of the most exciting advances in cancer. We didn't just want genome sequences, we wanted clinical and phenotype information. So we formed the health nucleus as a key way to get phenotype data, and also to try and change how healthcare is practiced. Today it's reactive. You wait till you get symptoms and you go see your physician or the British Medical Service, and they try and sort out what those symptoms mean. In contrast, we're trying to predict risk in advance, do early detection, and either eliminate the diseases or find ways to treat them. In the US, and I think the statistics are similar for Europe, if you're between 50 and 75 years of age and you're a male, 30% of you won't reach age 75. If you're a female, 20% of you won't reach age 75. The risk of dying, about a third in each case is from cancer, and another third from heart disease. The last third is from everything else. So just looking at cancer and heart disease, if we can detect those early, if we can predict them, if we can prevent them, it can have a major impact on people's longevity. We do a variety of tests aside from sequencing the whole genome, the microbiome, and the metabolome. We have a GE3T MRI where there's totally new protocols called restriction spectrum imaging that basically looks at the water differences in tumor nuclei versus surrounding tissue, in blood vessels versus surrounding tissue. So without any contrast media, we can actually tell the difference between a tumor and normal tissue. Some of the leaders in this field, David Caro, who does our radiology, showed that you can actually detect and diagnose prostate cancer with just the straight MRI imaging with this new differential imaging without contrast dye or without biopsies. These are the kind of images we get from an 8-minute MRI of the vasculature of your brain. You can see it shows up very brightly. This is not injecting any media. This is just looking at the water differences in the vessels versus the surrounding tissues. These allow us to easily diagnose things like this aneurysm in a relatively young woman. Normally the way these are found is they pop and you either have a massive stroke or early death. Now this is very treatable with a number of different approaches. We can also diagnose very readily things like Alzheimer's disease. There's a set of algorithms called NeuroQuant developed here at UCSD that measure precise brain volumes in 20 different brain regions. If you see that yellowish area there, that's the hippocampus. You can see how large it is in the normal brain. And in the Alzheimer's brain, you can see it shrunk with big areas around it. It makes it very easy with quantitation to detect even minor differences in this. And we can detect differences in people's brains up to 10 years before they show the first symptoms. If we combine that with genomics, we can sometimes predict it 20 years in advance before their symptoms. And clinical trials are going to start early on, perhaps just with this kind of information, where we can know in advance that you're likely to get Alzheimer's disease. And the trick is to try and prevent it, not seeing if we can regrow 30% of your brain at some late stage. We also find a range of tumor types. This is a cavernoma that we found in a 64-year-old male. Everybody that goes through the HELP nucleus are completely symptomless. They think they're completely normal. And we're making major discoveries in about 40% of them. This is sort of a blood vessel tumor that can cause massive strokes from bleeding. This one, I don't know if you can see, if you look at the top of the brain, there's that walnut size raspberry tumor. That most of these are not cancerous, but some of them are, but they can all bleed into the brain. So these people were completely unaware that they had these, and certainly if you had one of these, you wouldn't want to be a soccer player. We can readily find tumors now. This was a male in his 50s. It was found right under his breastbone, a 5-centimeter tumor. And you can see on the right with this differential imaging, the tumor lights up like a light bulb in it. Using robotic surgery and our MRI imaging, this tumor was removed a week after we discovered it, and it had just started to penetrate the surrounding tissue. So if he waited 6 months more, he would have moved into a different stage. As it was stage 1. He went home totally cancer-free and needs no further treatment. If he waited 6 months and it penetrated the tissue further, he would have had at least 2 years of chemotherapy and probably less than 5 years to live. So early detection in cancer is the way to completely eliminate the disease. With our comprehensive cancer program, we usually see people after they have stage 4 cancer. The other thing we get off the new MRI imaging is we can get exact quantitation of your muscle mass and different types of muscle, your peripheral fat content, and your visceral fat content. So visceral fat is the important thing for your health and longevity. We can actually measure the exact amount of fat you have in your liver. So less than 4% is normal. We're finding people with high 30% fat that was completely unaware of this. Eventually, they usually need liver transplants to survive. The only thing that changes your organ fat is calorie restriction. I was on a 30-mile bike ride with one of the world's experts on organ fat, and halfway through he said, exercise doesn't get rid of organ fat. And I said, you couldn't have told me 15 miles ago. You have to have calorie restriction. Obviously, exercise helps with calorie restriction, but it's dynamic and it's reversible if it hasn't gone too far. We measure multiple cardiac parameters. This is 4D echo. You can see all 4 chambers of the heart very accurately. We can look right down the valves. We can measure Doppler shifts to see the tiniest amount of regurgitation. Using these techniques, we found 4 people with greatly distended aortas that can get blowouts like a tire blowout. So they've gone in for corrective measures. We measure hearts just with a small device that you just wear as a skin patch that measures your cardiac rhythms for about 2 weeks. About 3 people out of the first 100 had episodic atrial fibrillation. I think you all know that's you get clots when your atria is not beating normally, and that's the #1 cause for stroke. So these people were put on anticoagulants, not knowing that they had these. And these are just preventative measure findings from these simple measurements that people didn't know they had. We're obviously trying to make extensive genotype-phenotype correlations because we have the genome, we have all these measurements. Some of these are very straightforward. From the MRI, we can find polycystic kidney disease, or we can find polycystic liver disease, and it's easy to find the genes associated with it. But gene changes give you probability statistics. They don't give you yes/no answers. So we're trying to find when they correlate, and perhaps you can still have the gene associated with polycystic kidney disease and not have the disease, which means you have other genes that help prevent the disease. So we're trying to understand and discover these preventative healthy genes as well as the ones that people associate with disease. We have a machine learning group. I hired Franz Ock out of Google to head that up. It's in Mountain View, California, where we're trying to predict all the different parameters we can just straight from the genetic code. For example, we can predict your height, your weight, your BMI, your eye color, your skin color, your hair texture, your hair color. And now we can predict your face and things from your voice. So just from a digital recording from your voice, we can predict accurately your sex, your age, and your height. And these same digital recordings correlate with physical parameters in the face of your face. Most people don't know it, but you lose your sex chromosomes as you age. Men lose their Y chromosome, women lose X chromosomes. And you can correlate just by measuring the genome and the amount of sex chromosomes in blood, for example, to correlate that with aging. Also measuring things in the metabolome, and with our sequencing, we get the most accurate measurement of the telomere length, and it does correlate very nicely with age. 15 years ago, if you asked me, could we predict your age from the genome, I would have said absolutely not. And I'm surprised that we can, but we're within plus or minus a few years now and being able to predict your age just from a DNA sequence. We went after face. We set up our own 1,000-person clinical trial where we took 3D photos of their faces, measured a number of other things, and their genomes, and took this into machine learning. This is just shows you with mixed ancestry. Here's a 40-year-old woman you can see with a very mixed ethnogeographic background. The top is her actual 3D photo. At the bottom is straight predicted, just straight from the genetic code. Our software has improved substantially since the first version. Here's the 3D photo of the subject, and here's the computer prediction straight from the genetic code. Here's another version. So while we have accurate eye color, we don't have the graphics yet to insert that in these, which makes the predictions look even more lifelike. The ethnogeographic background is one of the easiest parameters to predict. So you can see it's getting better all the time. When we combine all these features, we can readily tell 1 out of 10 just from the genome. And as the data sets improve, we think the predictions will keep getting better. We're combining all this. It's a huge amount of data. Just from ACs and Gs and Ts, we have over 3.4 petabytes of sequence. Indexing the entire internet in 2014 took only about 12 to 14 petabytes. So you can see the amount of data is massive. But we can reduce complex brain MRI images to a table of densities of different regions. We combine all this, we create 3D avatars of people. We combine all this in a report we give you back. If you print it out, it's a couple hundred pages. We give it to you on an iPad, and your iPad helps you walk you through your information. Look, these are the earliest days in this field. The predictions we're now able to make, having tens of thousands of genomes that are extremely accurately sequenced, is blowing our minds every single day here. We're having trouble writing up the scientific results fast enough. But I think this next era, the next 10 years, we're going to learn more than we have in all previous history about ourselves and about our genetic code. Thank you very much.
And what you may not know is Dr. Venter was watching you as you were watching him. He can see us. Dr. Venter, David here from Wired. I guess my first question is, how soon can you give me an appointment, doctor?
Well, it's $25,000, so as soon as you want one.
So we are on one of those exponential curves of the falling cost of sequencing the human genome. I think when you sequenced yours, we were in the $100 million region. Now we're falling towards a few $100s. I imagine for this level of sequencing to be a regular part of health service, prognosis, diagnostics, it's going to have to come down in price a considerable amount. How far away do you think we are from a time when it just becomes another bearable cost for the healthcare industry?
There's no reason it shouldn't be there now. So we can sequence the entire genome accurately for around $1,000. When you look at last year, BRCA1 and BRCA2 tests, they were charging $5,000 for those single gene tests. That's been part of the clinical picture now for a while. So we can get the accurate sequence of every gene in your genome for about a quarter of the price of understanding your BRCA1 risk. So there's no reason that shouldn't be part of the medical picture now. It will lower healthcare costs substantially by being able to make advanced predictions instead of waiting till you get sick to find out what's wrong with you. So it should be part of every medical system currently. Medicine doesn't change rapidly, as you know. It's going to be forced to change by the kind of data that we and others are generating so that people will realize they can't afford not to have this information.
But you are asking a lot of vested interests to kind of tear up their business model. I mean, the insurance sector, the pharmaceutical sector, how much resistance are you seeing now from the big power brokers?
Well, it's changing. I think you saw last week, I was over in the UK for our press conference with AstraZeneca announcing a 10-year deal where they're going to literally try and work on 1 to 2 billion genomes, having it affect every part of drug discovery, drug development, and clinical trials. We announced last year in the UK and in South Africa doing an exome for $250 as part of Discovery Health. So insurance companies are not only the leading edge of them, are definitely embracing these approaches. We're talking right now to every major life insurance company in the world. Your life insurance company actually wants you to live a long time. So the two of you have something in common. You're betting that you're going to die. They're betting hoping you're going to live a long time. Genomic data is going to allow you and they each to hedge their bets. They can't afford not to be in this business.
It's one thing for the trained clinical expert to have access to all this data. It's another thing for the untrained consumer to be given a data set, to be told, well, you have an X% chance of developing Alzheimer's, developing these other conditions in Y number of years. How much do you worry about the lack of education in terms of individuals, untrained individuals, interpreting their personal genomic health results?
Well, we don't leave it up to them to interpret themselves. We provide all the data back through our physicians, ideally through their physician, and through genetic counselors. I actually think that the average MD is harder to educate than the average person. And most people will be educating their physicians with their data as they do with information now off the internet. So medical tradition is the hardest thing to change, but it is complex information. In contrast to most people, when they do a genome report, like Illumina gives a 16-page report. We give a 500-page report on your genome, and it is a lot to understand. We try to help you understand it. We're building online tutorials and other means to do this, but it's going to be essential to understand this information, to understand how to change and protect your own life. So we're early on in this. Hopefully, with the use of the internet and online education, people will move well ahead of the medical establishment quite quickly.
Last question. We've got a room full of health policy practitioners, people who are investing VC funds in healthcare, people who are starting and growing businesses. What's the one action point you want to leave this room with, anybody who's watching us online?
Well, Genomics is no longer a zero-sum game. 15 years ago, when we were racing to do the first one, it was all dynamic and generated a lot of press attention. Now, with close to 8 billion people on the planet, the more people that participate, the better it's going to be for changing health policy across the board. I think you cannot underestimate the importance of the accuracy of the data and the completeness of it, and the ability to interpret it properly. Anybody can buy a sequencer now and do sort of half-ass sequencing of a genome. They don't have the ability with the data sets that we have to do comprehensive, accurate analysis. And as you mentioned, giving people accurate information is the key part. So investors should be investing in all kinds of ways, from educating the public to ensuring that the data is accurate.
Greg Venter, I think you're one of the few people that everybody in this room would agree is moving the world forward. Thank you very much for spending time today talking to Wired Health.